book with photographs of the cuckoo, and the book is called The Cuckoo, the Uninvited Guest. Definitely worth taking a look at that publication. Absolutely fantastic imagery. I'll give some photo credits and indication here whenever there's noteworthy examples, but this is absolutely fantastic. It's showing this coevolutionary struggle that occurs between the common cuckoo and the great reed warblers. And what's happening is that the cuckoo is grabbing out one of the host's egg, and her cloaca is positioned such that she's laying an egg right now in her nest. She's parasitizing this clutch, and both of the parents are doing all they can right now to prevent this from happening, but they're going to be left with this struggle, this challenge to be able to discriminate which of those eggs is the imposter, which of these eggs belongs to this female cuckoo. And the color of birds' eggs is really important for us to be able to for the bird to be able to make the good choices. And because now that we know that there's this particular line or this gamut of egg color gamut, we can actually find pigments that match the brown and the blue, and we can use admixtures of them to recreate that variation that occurs in nature and start experimenting. And that's what we've been working on. And it's very important that the bird has the ability to make these kinds of decisions appropriately, because she might be left with an unrelated and very large and demanding mouth to feed. And this is what you're going to see in these images. So the cuckoo will eject the host chicks, the foster siblings are removed from the nest, and she's left with this very exaggerated and energy demanding offspring that for which she gets no reproductive benefits. It's just a cost. It's a parasite. So this is known as brood parasitism. And what we believe is that the colors of the bird eggs is really central to this. This is the dastardly deed in action. You can see the cuckoo egg here, and then the cuckoos will hatch first as they're pre-incubated. And they have adaptations on their backs to be allowed them to scoop out and remove their foster siblings. And that's what you see here, meaning that they won't have to compete with any other siblings for this food, and can grow to really scary dimensions compared to the host mother. So there are all sorts of costs and all sorts of reasons why the host needs to be able to look at that egg or assess the eggs within their clutch and determine whether or not they've been parasitized. Because it's costly. It's costly in terms of this egg removal activity, which you see here. The cuckoo is swallowing the host's egg, meaning that's even harder to detect the deception. The host eviction, meaning she loses all of her potential fitness, and of course those energy demands. All of these reasons really mean that it's important for the host parents to be able to recognize and respond appropriately to this. And egg color is really an important feature for this. She, of course, can try to prevent the laying altogether by the cuckoo, and you saw that in that dynamic image. My focus has been on this egg stage, so we run egg rejection experiments. And of course, the chick stage is another potential area where the host can respond and recognize the parasite. Many hosts have adapted this ability to recognize eggs, and color has been a very important way or component that they focus on. And so as hosts have adapted this ability to be able to see and recognize and pay attention to those key pieces of information about the egg and detect those imposters, that is imposing a selective pressure on the parasites themselves. And so when the parasite egg escapes detection because it appears like the host's own, well, in many cases, the parasite may adapt eggshell mimicry. And so what we see over time is that in many contexts, host response to the cuckoo egg is removing genes from the cuckoo gene pool that are mismatched, so not producing good egg matches, and leaving those cuckoos who produce the best, most exquisite matches to the host's own. And that drives for greater discrimination ability in the hosts in the next generation. And this creates a feedback. And we refer to this as a coevolutionary arms race, where what we expect to find is that in those hosts that have the best ability to discriminate cuckoo egg from their own, they will have the be parasitized by cuckoos that have the best match. And so really it's the host decision making, the host recognition that drives this mimicry and drives the parasite phenotype. And so this can occur in many contexts. I know I've been talking about the common cuckoo, but what we're looking at here are sets of eggs between the cuckoo finch and the tawny flanked prinia. Many other systems have engaged in these types of coevolutionary arms races to the same end, where they have exquisite mimicry and well adapted recognition abilities. And so what we're expecting is that as hosts adapt an improved rejection rate, so meaning that they get better at recognizing on this end and poorer at recognizing on this end, we would expect that the difference between the host and the parasite should decrease. So we're expecting this negative relationship. And Stoddard and Stevens were one of the first to apply an avian vision model to this, and they found just that. Those hosts that had very poor recognition ability didn't have well adapted or really mimetic cuckoos, but as you sort of improved your recognition ability, the match between the cuckoo egg and your own became much more exquisite. This fits well with our understanding of coevolutionary arms races and coevolution in general. But what about all of those different parasites that don't appear to adapt mimicry? How can we explain those kinds of responses? What you're looking at are as nest of a Kirtland's warbler with a single brown-headed cowbird egg in the nest. Now, it might be simple for you to recognize which egg doesn't belong, but it's an extremely challenging task for these warblers. And we, in these types of situations, where the host does not actually respond, well then of course the brood parasite doesn't have to adapt and match. However, the brown-headed cowbird exploits a wide range of hosts, many of which can detect their eggs. And so you'd think that this particular parasite should be impacted by the decisions of their hosts as well. And so why don't we have mimicry or adapting in some of these systems? Is it only because they're using all of these different host types? Well, I'll tell you a little bit about the brown-headed cowbird first and some of our perspectives on how their phenotypes are adapted. So they, in theory, should share some kind of relationship between the ways in which, with the cuckoo, in terms of how the host phenotypes are shaped, or the parasite phenotypes are shaped by hosts. But of course, the system is entirely different. The brown-headed cowbird was associated with bison. That's presumably where it's got its name, in the Great Plains, so sort of North Dakota range. And this is a close-up of a male. They can parasitize, unlike the common cuckoo, they can parasitize up to 200 different host species. And the real key difference between the cuckoo and the brown-headed cowbird is that the individual females will lay into the nests of various host species. And while there's still a lot we have to learn about the choices that individual female cowbirds are making, this ability to lay into a range of different hosts as an individual is really key to the fact that the cowbird's egg does not sort of go down that same road that the cuckoo's egg did, because it's got all these different hosts. You see some of these host species here, and some of them can recognize a cowbird egg immediately. Some are less skilled than this. So you'd assume. Sorry. Before I get to that of course the range of the cowbird has changed. We've transformed the landscape dramatically since settlement and agricultural development has sort of opened up the landscape and allowed brown-headed cowbirds to invade other spaces. And so nonetheless, many of the hosts even in these newer ranges, are decent with egg rejection and egg recognition. So you'd assume that there should be some selection on the cowbird phenotype. But how would it work? Well, the cowbird's phenotype in general is this generic white egg with brown spots. It could potentially resemble many different host species. It's a pretty decent match for the cardinal. Sometimes it can be quite difficult to tell the difference between a cardinal and a cowbird egg, but really it's unlike most of its hosts. It's just this white egg with brown spots. And so very unlike what's going on with the cuckoo. And we should be able to we should be able to understand how rejectors will select for the for the parasite's phenotype. With the same kind of logic that we applied very simply to the cuckoo. So how can we actually do this? So what we think is going on with this particular system is that there might be diffuse coevolution. And so diffuse coevolution occurs between groups of systems, so groups of hosts and parasites, let's say, and it could occur in mutualistic relationships or parasitic relationships. And in this case, there's selection from a range of host species or the host community that's selecting for the phenotype of the cowbird. And so to look at this, we've got to create a phenotypic space that fully appreciates the variation that we see in birds' eggs. Now, if you remember, we had that color gamut, which is that line throughout the visual space. This underappreciates some important aspects of color or appearance, let's say. And that key portion of appearance, particularly for birds' eggs, are white and lighter eggs. So to differentiate between the darker pigments and the lighter pigments. The tetrahedral color space I showed you earlier, with which there was a nice line through that space, only looks at colors. There's no brightness information. So we need to be able to add that in because the phenotypes of bird eggs are roughly white and blue and brown. And you could see such a space here, and I've got various host eggs plotted here forming somewhere in this space. And you'd imagine that if they all selected for the cowbird egg equally, that the cowbird egg's phenotype would lie somewhere in the middle because each host is sort of pulling that parasite phenotype towards its own with every egg rejection. So could we develop a simple model that allows us to look at coevolution or pairwise coevolution of the cuckoo on one side, but equally can look at the behaviors and phenotypes of the host species of a parasite like the brown-headed cowbird and predict its phenotype? In theory, we should be able to come up with that. And I'll sort of walk you through what this graphic is. This is the difference in the brightness. So again, in that phenotypic space, and the differences between the host and the parasite in color. And so in this particular case, we have unexploited hosts in the area for the female cuckoo. And the adapted phenotype, if the host rejects, will eventually be mimicry. So it's sort of pulled in this direction and no other host is pulling it away. But now when you look at a system like the brown-headed cowbird that has many different hosts, all of which have different colors and different recognition abilities, which is what those numbers represent. Now some of them are going to be pulling to different degrees, and different hosts are going to be pulling. And so right now I just have these red hosts that are brighter or darker or browner or bluer that are pulling to various degrees, and we should be able to predict what the phenotype of that parasite is. And the question is, can we do this? So can the same kind of mechanism under diffuse coevolution and imperfect mimicry work to describe this perfect and imperfect mimicry? This is sort of the way in which they're being pulled to various degrees, the length of that arrow being the strength of that pull. So we would basically predict that the cowbird phenotype should be predicted by, if it's predicted by diffuse coevolution, the colors and rejection abilities of those hosts should select for the cowbird colors. So we constructed a model that lets us predict the cowbird phenotype or as a weighted centroid within that space. So we could basically put in the phenotypes of the hosts and how much they pull that phenotype and predict where that cowbird would be. We collected data on 43 different host species from museum specimens and resampled these potential host communities, basically shuffling through the hosts so that any particular draw was of different cowbird population that might be subjected to a slightly different host community. And each time we predicted what the cowbird phenotype would be, and then later we can compare that with the cowbird phenotype. And if it's being selected by diffuse correlation, the cowbird should be well predicted. And what we found is that those host communities almost always predicted, up to 98% of the time, predicted the cowbird phenotype. And this is that space, the actual data of the 43 different host species. The concentric rings here correspond with the just noticeable differences, where the first ring, anything in the first ring is not even perceivably different, even in clinical sort of conditions. The second ring, two just noticeable differences, most likely won't be noticeable in the field, sort of in real life kind of scenarios. And the third are similar, but the bird likely would tell the difference between them. All this cloud of points was all of our predicted cowbird phenotypes, all within that 2 JND mark. And so these are the predicted cowbird phenotypes, and the actual one was here. So they were all well predicted. The other key point was that hosts typically were bluer than the cowbird. And so that could be important. So this is the red ring I was referring to here on this slide. So basically all of the predicted phenotypes were well predicted. And this has some important implications. It implies that hosts select for parasite phenotypes. So even in this context of the brown-headed cowbird, the hosts are playing some role at governing the parasite phenotype, much like the hosts do for the common cuckoo. And it might also explain imperfect mimicry in other kinds of systems. So other kinds of cuckoo models even have situations where there's two exploited hosts or hosts with particular phenotypes, the polymorphisms, and the parasite's right in the middle. And so it might be able to explain those contexts as well. But another interesting question is, why do hosts tend to be bluer than the cowbird? How pervasive is this? We've gone on to address how hosts are responding to color variation. And one of the reasons why this might be is how they respond to parasitic egg colors. We've used the kind of data we've been talking about here with perceptual spaces and to model colors of natural history specimens and construct colors that we can use and test in the field. And this is data that we applied to a common blackbird. What you see here are the colors of the egg models we constructed, and in black we see the color of the host zone. So we use that museum data to actually come up with good paint mixtures that allow us to extend the entire range and ask questions about how they might respond to it. We print experimental eggs, paint them, measure them, find host nests, deploy the egg models, and assess what birds do. And we do so by looking at eggs that are very different in terms of blue and sort of similar or very similar in terms of blue and similar in terms of brown and very different in terms of brown. So hundreds of egg models that vary in the degree of similarity in either the blue side, the blue direction, or the brown direction. And if you recall, we would assume that if hosts are just responding to color variation, it shouldn't matter if it was different in blue or different in brown. So we should see these U-shaped curves when we look at the probability of rejecting any particular color. But interestingly, this doesn't happen. Many hosts appear to respond more strongly to brown colors than they do to blue. And there's actually some interesting potential cognitive and perceptual mechanisms that could explain it. But regardless of the underlying reason for why this is happening, hosts do appear to behave in this way. And that might explain why hosts themselves end up on this side of that spectrum. So hosts all over the world sort of responded in this way. And and we've tested this in Europe and North America, South America, and basically all of them have responded in that way, but all of them have bluish eggs, which is an important point that maybe we could discuss in the question and answer period. So the question is, how are these colors shaped? Clearly, these coevolutionary dynamics play some role, but the parasites themselves are also constrained by their environment. So they are going to be facing these same weather patterns, dietary restrictions, and other factors that are going to be impacting the colors of their eggs. And there's certainly going to be trade-offs between the kinds of traits they invest in and the kinds of phenotypes that get manifested. And one of those trade-offs is this balance between predation and thermoregulation. So birds have to be able to balance detectability of their eggs and the risk of overheating. You're looking at a really fantastic photograph that was taken at a hunting lodge, and the idea sort of goes like this. These bright white eggs can draw attention of potential predators. However, those bright white eggs are very useful in that they reflect a large proportion of the instant light, meaning that they're not going to absorb that and overheat. And so eggs have to maintain this strict thermal temperature within these strict thermal limits. White eggs in hot climates can reflect a lot of heat, but also can draw unwanted attention. However, depending on where you live on Earth, you could have the opposite situation arise. So if you live in the coldest regions on the planet, this trade-off, this life history trade-off, actually sort of flips and becomes this synergistic benefit. So in these particular places, you have the dark brown eggs potentially providing the benefits in terms of crypsis or lack of detectability, and also can absorb radiation where you need it most in these colder environments. And so sort of you tip the scales as to of this trade-off depending on where you live on Earth. And really, museum specimens are perfect for answering this kind of question. And first, to tackle this challenge, we need to look at to quantify variation, or how well bird egg colors relate to their thermoregulatory ability. And we needed to do this because many of the methods that were available at the time were incomparable in nature, like, for example, coloring eggs with crayons or other kinds of techniques that might inform us of something about temperature with respect to color, but wouldn't really be comparable to natural egg. Or they were conducted and the information would be comparable, but it was within a single species that didn't have very much color variation, certainly not that full gamut of coloration. So to first assess this, we took internal and external temperatures of birds' eggs, and we just wanted to assess how egg color related to thermoregulation, considering that full variation in color that we see. And we chose chicken eggs as a good model system. They have blue, white, light brown, and dark brown variants, which you see in these fake nests that we constructed with one of our grad students, Phil Wasaki. We allowed eggs to cool from an incubation temperature under natural light. So they're light incident on the surface of each of these eggs, and you can see that one of the thermal images here and the layout of the eggs here. And I'm going to put on the screen two indicators of where the white eggs were and where the the dark brown eggs were with these icons. And I didn't really line this up perfectly for this video, but we'll sort of watch this and see it change over time. And the key point to look for is that those white eggs ended up cooling, turning blue color, much more so than the dark brown ones. Those dark brown eggs ended up retaining their temperature. And so we did the same kind of thing where we basically had the eggs at room temperature this time, and we put them into the full sun and allowed them just to heat naturally to see what temperatures they would achieve. When we did these types of experiments, we could see that there was fluctuations in temperature over time, and they responded to environmental variation like cloud cover. But the heat appeared to as a function essentially of pigment. And so those those darker, browner eggs tended to secure greater amounts of heat over this period of time. So in this case, these were room temperature eggs, and they nearly got to incubation temperature if you were brown. This is really important if you live in the Arctic and in relatively cold ambient temperatures. So this graphic here shows some of the results of this experiment, where we had the egg phenotypes from this particular experiment across time relative to the white egg. And one of the reasons we needed to do this was because different trials had different ambient temperatures and different conditions. But we always made our measurements for this series of experiments relative to the white egg. And that way we could look at how much heat a particular egg had with respect to that. And over time, you can see the accumulation of heat is roughly correspondent with the blue, light brown, brown eggs. So the brown eggs over a 25-minute period had accumulated, on average, 4.5 degrees more. We can look at this in terms of starting at incubation temperature or ambient temperature. And what we see is this nice linear pattern where, generally speaking, about 4 degrees more is secured by these dark brown eggs. So the other interesting point here is that this line right here, this dashed line, this was the same experiment conducted in a dark room. And there were no differences with how much heat they shed in darkroom condition. It was really the absorption of that pigment from the full sun that resulted in this property. And so this was actually done on a variety of species. We have chickens, duck, and quail. But this does show us that pigments and brown and dark pigments do have thermoregulatory potential and can be quite useful. And so our first investigations showed that those brown eggs will heat more rapidly, up to 4 degrees more than those white eggs, due to this incident solar radiation. And the question remains whether or not birds have responded and adapted to global variation in climate. So are those synergistic benefits that I mentioned earlier visible on a map? Can we see that effect, essentially that theory that we've laid out, on a map? So we turn back to that museum data set, the full phylogenetic diversity, and we also turn back to that phylogenetic space that you looked at, variation in brightness, essentially, and color on the other axis. And we can quantify these things across all these different orders and families and genera. And so eggs will sort of sort in these different places within this phenotypic space. And what we did is we did a random sample for each of the birds in our data set, our museum database. We simulated nests at a density of about one nest per 10,000 kilometers. And you could see that sampling on the screen. These are individual little points that are across the ranges of all these birds, some of them overlapping with one another. So whole communities will differ depending on where you are in the map. And what we needed to do was to sample this appropriately. So we laid onto this hexagonal grid that allowed us to look at unique communities that live in only that place and calculate the phylogenetically corrected, or the mean coloration, for this particular set of organisms that live in that particular place. And we did this over and over again, gridding the entire world with these hexagons until we came up with the average brightnesses and colors of the birds from each of these communities. And of course, the hexagons are a little smaller here, but you can see that this variation that occurs around the world, where there's darker, browner colors in the Arctic, and as you approach the tropics in general, you get brighter and blue kinds of colors. And I'm going to change this graphic slightly for those that might have trouble discriminating those types of colors, so you can sort of appreciate those patterns as well. Now, that's great. So now we've got this map that lets us assess what colors exist in those avian communities, but what about the climate in those particular places? So we ran a PCA of the mean annual temperature, ultraviolet radiation, precipitation, humidity to construct essentially two PC scores that captured that variation. PC1, solar intensity, so temperature and UV radiation really falling out on that principal component, and aridity, So precipitation, humidity on the other. But we can make this colorful map that essentially lets us quantify the variable amounts of each of those. And you could see distinct ecoregions presented on a map like this. And so the question is now, I'm actually going to focus mostly on this PC1. And we're going to look at solar radiation with respect to eggshell brightness on the left and color on the right. And there's a couple things to note. I've plotted out the latitude as blue to red. So the blue colors are really the far north and or south, and these are absolute values, and the red are the equator. And the patterns are pretty tight. That's that synergy that I told you about. We expected to find this synergy between the darker brown colors in the north that are beneficial for crypsis and beneficial for thermoregulation. And really, we sort of expected the opposite in the tropics, but all bets were off. I mean, the colors and the brightnesses were all quite variable, presumably because a lot of different selective pressures occurring by the tropics. And we could also parse this out by nest type. And this was one of the key, in my view, findings in this particular study. When we looked exclusively at birds that nested on the ground, and in the open, those patterns should be strongest. And so that's basically what we see here. The patterns with eggshell color were driven by mostly by those ground nesting birds getting browner in towards the north. And with brightness, they were attributed to both the ground nesting birds getting darker to the north and cup nesters getting darker to the north. This really was not significant, importantly, within cavity nests. So the question is, how can we expand this to really use museum data sets in in new ways to be able to address these questions? This was a pilot project done by Sela Cooper, which you see on the screen, that used the Shiraj form data set from the Delaware Museum of Natural History from old photographs that I'd taken at their egg collection. And we are able to measure the spectral sensitivity of our camera sensors, allowing us to quantify the same kinds of relationships that we saw before using the spectrometer, using very simple algorithms that allowed us to transform those colors into avian perceived colors. That tetrahedron, again, is shown from above, and there's some noise still in this data set, but that had to do with the camera being sort of not designed and the photographs not designed for this purpose. It was one of those opportunities that manifested itself due to the COVID situation we see ourselves in. We found new ways of using that color information. But there are lots of ways that we can transform how we are thinking about these kinds of data sets and how we use them. So the main findings are that egg colors are surprisingly limited. Diffuse coevolution might explain some of the variation that we see or how hosts are selecting for certain parasite phenotypes, and temperatures do drive the evolution of egg colors. And so we hope that and feel that these findings will help us reveal both patterns and processes in trait evolution. And integral to all of that has been the museum collections. So the questions remain: how are we going to integrate this sort of image processing and collect massive amounts of data on these phenotypes, and whether or not those phenotypes are likely that we discover are likely to reveal new and interesting patterns, and we have yet to see those discoveries, and so we're very much looking forward to tomorrow. So thank you so much for this opportunity. I'm going to be in the room with you guys. If you have questions before, I hopefully would have addressed them, and hopefully there'll be ample time to address any remaining questions you have now. Thank you so much.
Excellent presentation.
Thank you so much,
Daniel. Yeah, that was great. Thank you very much. So I know that you were answering some questions during the chat, and you have such a broad research background already. into egg coloration, and you presented a number of different packages of that research. And so, you know, we can go all the way back to the beginning and ask questions about that, but I thought it might be more natural actually to maybe see if there are questions about the last part and then work backwards, but we can also just see kind of what arises. But just to start it off, and people can maybe raise their hands or note otherwise, like if you turn on your cameras, everybody's welcome to, if you want to wave at us, and you can indicate that you have questions too. But I have a question, since you ended up with the conversation about the relationships between egg coloration and temperature throughout the globe. I would like to see what you think in terms of actual predicted coming climate changes. Of course, this is like a softball pitch, but really, like what do you see? What do you predict actually is going to happen over time to you know, some of the birds either that you worked with or that you've in general, you know, modeled?
Yeah, so my suspicion is, so we have yet to sort of study this. What I'd really love to do is study birds, particularly ground nesting birds with the darker brown eggs in the lower Arctic. And so that area is warming much more rapidly than many other places on Earth, and they've adapted for a very, very long period of time for those sort of colors that would have been quite adaptive in that location. They're the ones that I think, in terms of their egg colors, are going to be at the greatest risk of overheating. And so I think there's, if you sort of overlay the rate of change of climate change and the colors of those birds' eggs, you can get this like band of at-risk species. And it's going to be probably in the lower Arctic. That's probably where we're going to feel it most. And interestingly, there's a researcher here that works with longspurs and has sort of noticed that some of these species are sort of moving in almost counterintuitive ways. They've actually moved into these other kind of nesting sites that actually might that it would be counterintuitive to sort of I wouldn't have expected them to, but in that region, in the lower Arctic, I think that's going to be the most at risk. And I've talked to a few other researchers, particularly those in Australia. They have a very different perspective on heat and heat tolerance in birds, and so they're very much sort of thinking not about the lower limits. So I think about the lower temperature limits and making sure that you could stay warm. What they are focused on, because of where they live, is really don't overheat. And the very relevant point in these conversations that I had with Debbie Stewart Fox's lab, and so they're working on temperature work as well, is you don't come back from cooking, right? So many birds have adapted chill tolerance. And so while I think that's very important, and we can't get that nice band to the lower Arctic, if you exceeded your thermal limit, that's it for you. And many birds have adapted these chill tolerances. So I think that the surprising things will happen sort of in the lower Arctic, but I think that there's very different dynamics on what matters to you as a bird, depending on where you are. So those upper limits are real risky. So they're at severe risk just because they're already tightly sort of managing. The parents can't. There's a whole story about what the parents do. So the parents have adapted their foraging bouts such that they can keep track and keep their metabolic energy themselves. And I talk a little bit about it in the temperature paper. It's about how they sort of keep track and can both protect their eggs, but also secure enough food for themselves. And both in the Arctic, but also in those high temperature areas, they're at their wits' end. I mean, they're sort of have adapted to just get by. And so at either of those two ranges, I think that, we might see trouble.
So deserts, extreme deserts, and extreme cold areas, arid areas in general.
Mm-hmm.
Then,
yeah, I think so.
They're already pushed one direction or the other, but they're at their edges. Yeah,
I think so.
Makes sense. So can I open this up then to other people who might have questions? You're welcome to either unmute yourself or raise your hand. Francisco Magdaleno, you have a question.
Yeah, Daniel, I was wondering for that last part, what program are you using?
So the program that you saw there, I just used R. The algorithms are relatively simple. We do them in Excel too. Like we've got, I think we could do a lot of this in Excel as well. We use Excel, R, and Python, depending on the image data set. So Sela was new to research. So we started in Excel and then we moved to R. There are lots of ways. So, and we also have ImageJ codes that will help us. There's a really good tool set by Julian Troshenko that uses ImageJ as its basis. And that's a really good one to start to use. And for that very last image for the image analysis of the Delaware Museum, that was sort of an introduction to research, introduction to ornithology. And so we started from the ground up. So we just built the, you use some matrix algebra. There's certain steps and you could do them in Excel. And we sort of went through the steps in Excel, and then we built a simple model in R. Yeah, if you're interested feel free to write and I can, give you lots of details.
Because the ImageJ, I'm familiar with ImageJ, and then the Mika one. I think that's the one you're referring to. Yeah. Okay. So do you recommend Mika, or it seems like
a lot
you're
of
doing
people
your own coding?
Yeah, a lot of people like that. There's I would recommend it. Yeah, but we do most of our own coding. So some of the stuff that we want to do, it would be too fast to process via with the Mika toolbox. And some of the approaches that we have in terms of the hardware that we've been building doesn't interface very naturally with that system. We had, but if it works for you, I don't know if you've got it and it's working, it's very efficient. So I would suggest it from that standpoint. But once you sort of break down the algorithms you need, it's not too hard to do in another kind of language that works better for you. That's maybe has less. Mika's got a lot of extras in it, and so you might want to shed some of that and just write something very simple that can do what you need it to do.
Cool. Thank you.
Yeah, no problem.
Okay, does anybody have another question, either about this last section with temperature or about anything else that they'd like to bring up right now? You can just feel free to unmute yourself.
Well, maybe I'll just do a follow-up to the temperature and climate change question that Linnea had asked. And your focus is on egg coloration, and you mentioned behavioral changes, adaptations by the parents as well to increased heat load potential. What do you think about other selective pressures on eggs, like shape or size and shape? Is anyone looking at that? And how quickly might you see that sort of? I know there are probably strong physiological constraints on that too. So just thinking about the value of egg collections and whether there would be enough data to actually go back and look at changes in any of those parameters over the last four or 5 decades.
Yeah, that's a really wonderful question. Cassie Stoddard did some really fantastic work on egg shape recently, and I think that question of whether or not there's adaptive shifts in egg shape and some of these other features is an interesting one. With respect to linking that back to climate change, not talking about colors per se, but talking about these other features, clutch size is a very important one as well. And there's some flexibility there in terms of maintaining your thermal mass. And so clutch size is a very important one that we haven't yet considered when we were looking at these thermal properties, and very, very important. And then sort of extending that phenotype beyond, that extended phenotype beyond the nest structures themselves and nest placements themselves are really, really important. I'm not sure, it would be fascinating. I'm not sure how egg shape itself might interface with conductive cooling or anything of that nature, which you might be implying, but that would be really exciting. I don't think I've heard anyone sort of suggest that, but certainly the evolution of egg shape is, particularly the rapid evolution of egg shape, even outside of the context of these thermal questions, is really a neat potential direction. Yeah, thanks.
Surface area versus volume is the parameter of interest.
Mm-hmm.
Right, that's what I was thinking of.
And then aside from even shape and size, I was just going to jump in and just, mention again that there's all the issues in regards to shell thickness and impacts from contaminants and various environmental pollutants still on eggshells, which is still a vital area for research and very useful for monitoring. We think, Daniel, you had a paper in which you actually looked at that and looked at the possibility of just changes in eggshell color as an indicator, a bioindicator for the degree of contamination in the environment. So there's some really other interesting ways to use, eggs and color and information. And then there's just the properties of the shells themselves, characteristics of the matrices, characteristics of the pores, and etc.
Pore size versus thermal emissivity. Things like that. Oh, I had a question from earlier. If I didn't just lose it, saying that, oh, okay, when you were doing the test with real eggs, the chickens and ducks and quail, did you look at emissivity as a function of color?
So we used the standard, and I forgot exactly how we calculated it, but we calculated the emissivity for each of the egg types, but we didn't. So we could have gone into much greater detail with the emissivity and allowed that to fluctuate, but we did have a specific emissivity that we use that we calculate for those samples. But there's a lot more we could do with respect to that.
Getting back to the environmental question, the question is, for example, black is very emissive, and so it cools down rapidly. And so it might be counterintuitive that a brown egg might actually lose heat faster, and it might go the other way.
Right. Yeah. Interesting. So when they were in the dark room, so if that were true, when they were in the dark room, that they should cool at different rates. Yeah. But they all cooled at the same rate. And so what we suspect is that that darker brown is just absorbing more heat, and so it can't cool down as quickly because it's absorbing constantly, where the other one is more or less shedding heat.
Basically says they all have about the same average emissivity.
Yeah. So yeah.
Because they're not actually black, they're still
right.
Brown.
Well, it's black and the type of material.
Aren't brown eggs white on the inside?
Usually,
there are a lot of things that fold into this, so it's.
Interestingly, and with respect to almost black, I mean, the concentration plays a pretty big role in the color of the egg as it photo oxidizes. So like an emu egg, if you see like a poorly stored emu egg, it goes through these phases of almost going black from that nice color it is when it's laid to being nearly black. And then if you see one that's really been left out too long, then it goes white again. And so if you look closely at the pores when it gets into that nearly black phase, and it'll show up this way in books a lot of times as sort of black, and to some extent catbird eggs will do this too, because they're very dense pigments. When they photo oxidize, there's such a high concentration of biliverdin that actually darkens, whereas another species like a wood thrush or something, it's got much lower levels, and when it photo oxidizes, it initially decreases. So there's some really cool dynamics there with change over time and oxidation.
Which makes it really hard to predict what's going to happen with climate change.
Yeah. So can we go back to the question just in the interest of time here? And Bruce, I hope that at least addressed your question a little bit. Okay. I just wanted to go back to where you kind of did a leading question, Daniel, during the recording, during your presentation about the blue eggs, when you were talking about diffuse evolution. And what happened with either hosts that have blue eggs or cowbirds trying to match those blue eggs, and you said, okay, maybe we could talk about that a little bit more, because it was kind of an interesting finding from that work in particular. Yeah,
so there's some really interesting perceptual properties that go on with color discrimination. So if you try to discriminate two colors on a very similar kinds of backgrounds, they don't have to be very different for you to see them. But if you start to discriminate two colors that on a very different background, even if they're the same perceivable difference, it's actually hard to tell them apart. And you could do this with a construction paper and just lay this out and take a look at this. And I think that simple property, this chromatic adaptation, it has to do with the way that our eye has been adapted to compensate for these kinds of differences. So what's happening is when you've got this big brown nest with which you're viewing these blue stimuli, your eye is basically adapting to that background and up or down regulating some of the photoreceptor sensitivities. And as a result, those differences that to me in these models and in the lab are equally different to the differences in the brown are not equally different in practice. And so that while maybe I should have increased the J&Ds of how different those blue eggs actually were. In practice, the birds are actually engaging with these kinds of discrimination tasks. And that could sort of shape and push evolutionary processes in certain directions. And so there's that whole range of things that I don't think really anybody's engaged with yet, those kinds of questions and how that interfaces into the coevolutionary arms race. And as a result, that's like one side of things. And the other is if there's systematic biases towards rejecting certain colors, you would expect certain hosts, particularly those in the north where blues are not as common, to have bluer eggs. So hosts in the north, should have unique kinds of eggs that should be able to recognize imposters. And if most birds, and there's a few reasons that we think this might be true. Some birds have this predilection to sort of behave in this way that has more to do with the way that their brain and the processing of those color information works. And so if they consistently do this and remove certain kinds of colors, you're always going to want to stay on the other side of your parasite. And so there are a couple data sets that we're playing with that are sort of exciting, that sort of show that there's this shift towards these certain spaces and suggest that some of the interactions they even talked about with like the coevolutionary dynamics don't operate the way that we think that they do, which is kind of exciting.
That's very interesting. Yeah, very interesting.
Thank you.
Does anybody else have another question that you'd like to slip in here before we let Daniel go? Because it's 3 hours later where he is.
It's Friday night.
Okay. Okay, I think we're going to let you go then, Daniel. But thank you so much for the overview of your work. And the recording of Daniel's talk will be available on our website. We also can send you a link if you don't see it. We're happy to, share additional information with you. Daniel, also, I'm sure that if any of these wonderful folks actually had questions for you that they didn't want to share tonight, that they could reach you. Do you want to give your George
Yes.
Mason email?
Yes. Yeah, definitely come and play with eggs with us if you come out this way, or talk birds. So feel free to write. And yeah, thank you so much for having me. This has really been a treat. Thank you.
Yeah, thank you very much. And there were several people who were on tonight who are from various collections or from, like Susie, if you don't mind me saying, so also from zoos and who've worked with breeding birds and their eggs for years. And so I know that they were taking this in and probably will have follow-up questions for you.
That's fantastic.
Daniel has a lot of papers. So also if you're interested in reading his literature, it's on the ORCID site, or if that's how I'm pronouncing it. Is it ORCID? ORCID.
I've never had to say it.
But you can also just search for him, and he has publications, and I'm sure you could ask for PDFs, but he's done an amazing amount of work. So again, thank you very much, Daniel.
Thank you so much.
Thank you. Lovely to have you. Have a wonderful weekend, everybody. Thank you for coming.
Thank you.
Okay, bye-bye.
Nice to see you.
Bye-bye.